And thanks for adding your locations in the chat. Feel free to continue adding your locations. We love to see where everyone is joining us from. Typically, at this time of the day, it would be mostly Europe, Western Africa, US, Canada, South America. But wherever you're from, even if it's the middle of the night, welcome. And we'll start in just a moment.
Germany, that's our neighbors in the Netherlands.
Excellent. Were you able to make it to the ISI WSC last week? No? All right. Hello, Belgium. Benin. Good afternoon, Benin. And we have some others joining. As others are joining, it's 2 minutes after, so I will just begin our introductions, our official introductions. I am Peter Doherty, Executive Director for the International Biometric Society, and I'd like to welcome you all to the October Journal Club. These sessions are complimentary to all IBS members with a primary purpose of providing a platform for individuals to present worthy papers in a public setting and also to widen the scope of understanding of these papers. Additionally, we want to provide a networking opportunity for IBS members through this journal club, and that opportunity extends to the IBS online community as well. We'll talk about that later. But for now, I have a few general housekeeping comments as we get started. This session is being recorded, and it will be available for replay early next week. If your microphone is not yet muted and you're not officially speaking, please mute your microphones throughout the session. There will be an opportunity to ask questions during the Q&A period at the end, so you can familiarize yourselves with the features at the bottom of your screen for the chat and also the raised hand feature if you choose to use that. You may also ask questions via the chat feature, and we will address them during the Q&A portion. And that is it for today. Happy Friday, everyone. And I'd like to hand the program over to Jen Lee, the host for these journal clubs. Jen, the floor is yours.
Thank you, Peter. Yeah, thank you, Peter. And welcome everyone to this journal club session. Today, we're very pleased to have Dr. Judith Locke joining us as a presenter. So her paper was recently published in Biometrics entitled Learn As You Go, or LAGO Trials, Optimizing Treatments and Preventing Trial Failure Through Ongoing Learning. So apparently the big crowd speaks to the volume of the importance of this topic. So Judith is a professor and associate director of statistics at Department of Mathematics and Statistics at Boston University, and her research program mostly focuses on causal inference methods and survival analysis in HIV/AIDS-related problems, mainly using observational data. And she is also working on this LARGO clinical trial designs, where composition of the intervention package changes over time as earlier stage outcomes becomes available. We are also very glad to have Dr. Lu Tian, a professor of biomedical data science and statistics at Stanford University, as our discussant today. So Lu's research primarily focuses on survival analysis, semi-parametric modeling, resampling methods, and personalized medicine. So all participants are encouraged to engage in the discussion and ask questions. As Peter said, you can put your questions in the chat, or you can leave toward the end when we'll have an open floor for Q&A. And we'll also monitor the chat over the talks. So without further ado, let's welcome Judith. So Judith, please feel free to share the slides, and the floor is yours.
Thank you so much, Jen Lee, for the nice introduction and for organizing this and for inviting me to speak here. I'm always very happy to talk about Learn As You Go, LAGO. So Learn As You Go, LAGO is a way to adapt the intervention while the trial is already ongoing. Give me a moment so that you can see everything. No, that was not what I wanted to do. I want to take that. This, yes, now you can actually see the slides. So thank you so much for having me here. I'm always very happy to talk about Learn As You Go LAGO. At the moment, I think this is as important in my research area as it is the causal inference. And the reason is actually that it is quite similar to the first days of causal inference, because people, especially statisticians, have lately been refusing to analyze data of trials where the intervention has been adapted over the course of time. And this came out of a focus group that was led by Professor Donna Spiegelman on her big DP1 grant, probably the first grant of this type that was awarded to a statistician. And she was asking around what are the most pressing issues nowadays for statisticians to work on that are really pressing for practice. And this was one of the things that came out, because lately there are more and more large-scale implementation trials going on, given that there are a lot of implementation strategies needed for all these medicines and strategies that have been proven to be effective, but that are still not being rolled out to the full extent to which they could help the public health. So these trials are ongoing, and often the intervention consists of several components. And over the course of time, these components are often adapted because people feel that if the strategy is not working well, then they should adapt it in order to save the trial. But then the statisticians on board were often refusing to analyze the data because, hey, you cannot adapt the intervention in an ongoing trial. So what we tried to do as statisticians was to try to figure out, are there maybe conditions under which we can adapt the intervention in an ongoing trial so that the statistical properties of the estimators at the end are still okay? Things like consistency, asymptotic normality, bootstrap works, and also things like, can we test for an overall intervention effect, even when the intervention gets adapted over the course of time. So that's what we've been working on over the past few years. Our first paper appeared on logistic regression in the Annals of Statistics, and what I present here is our second paper that was published in Biometrics. The person who was really leading this effort was actually Ante Bing. He is a former PhD student here at Boston University. He just started working on Amazon, so he is still working on his publishing his second and third papers, but he led this work. So a lot of the credit for this presentation goes to him. This is also joint work with Daniel Nevo. He also led the first paper on LAGO. He is a former postdoc of Donna Spiegelman and is now working at Tel Aviv University. Learn As You Go LAGO seems to close a gap in what people actually want to do in practice. It has been implemented in a number of ongoing clinical trials now. Examples are Pulisa, Uganda, MEPIT, Nigeria, and TaskPen, Zambia. And they are large-scale implementation trials with complex interventions, and their intervention composition is also adapted over the course of time. And they are in people living with HIV, primarily focusing on cardiovascular outcomes. If this is all you take out of this presentation, then I'm already happy. So adaptive designs are popular in clinical trials and other randomized studies, and the goal is to modify a study while it's ongoing without jeopardizing the legitimacy and soundness of the trial. And the FDA defines an adaptive study as a clinical study design that allows for prospectively planned modifications based on accumulating study data without undermining the study's integrity and validity. Examples are sample size adjustment, removing treatment arms, and changing the target population. To the best of my knowledge, it doesn't include so far adapting the intervention component composition. So I am presenting today on large-scale intervention studies with a learn-as-you-go or LAGO element. So that means we are going to adapt the intervention in an ongoing trial. We are going to do that in a very systematic way to not undermine the study's integrity and validity. An intervention could be a treatment, a device, a new way to organize care, or the implementation of an intervention or intervention package. The current LAGO trials are of the latter type, where it is implementation studies. Typically, this intervention will be composed of various components, and possibly some of these are continuous. And a priori, people have an idea about what will work and what will not work, so they will recommend an intervention package for the first stage. But this is usually not certain, and finding out what is the best possible intervention package is actually one of the study goals. An example that is not a LAGO study, because it came before, but at the moment we don't have data from LAGO studies yet that we can share. The first LAGO studies have closed data collection, but the data is still being cleaned. So I will illustrate the LAGO design with a Better Birth study. In the Better Birth study, the intervention was to encourage the use of a checklist to ultimately improve the safety of mother and the newborn child. And the details on how to encourage the use of the checklist, so the precise intervention, changed over time, even the recommended version. This Better Birth study was taking place in Uttar Pradesh in India, and the outcomes have been published in 2017. It is actually a very large failed study in the sense that there was no significant effect on the primary health outcome of the final randomized clinical trial. There were several package components in the Better Birth study. The first one was the intervention launch duration. Then there was the number of coaching visits. There was a data feedback form. There was a Better Birth champion. There were several components. And the study began with a series of pilot studies to find the optimal intervention. And during these pilot studies, there was a process outcome collected, such as was each of the Better Birth checklists items actually carried out during each maternal birth, during each childbirth. The first pilots didn't include health outcomes, so I will illustrate LAGO here with outcomes being process outcomes. The randomized clinical trial that was the end of the Better Birth study also collected health outcomes, which I will ignore here because I want to illustrate how LAGO could have been used based on the data from all the three stages. The goal of the analysis of a LAGO study is often to find the optimal intervention package and also the effects of the intervention package components. So maybe we want to know how the mean outcome, for example, the mean proportion of items on the Better Birth checklist that is carried out during a birth, depends on the intervention package X. Let's say X consists of both the launch duration and the number of coaching visits. I have here only two intervention components because in Better Birth there was a lot of collinearity. What we may want to know is the optimal intervention package that has a success probability or a proportion of Better Birth checklist items that is carried out of at least 80% for a certain intervention package. So X here is the launch duration and the number of coaching visits. We're going to try to make sure that at least 80% of the Better Birth checklist items is carried out during each birth. This is not enough to have a unique solution to the optimal intervention package. So what we will do that while minimizing the cost of the intervention package and also subject to natural constraint of X. For example, it doesn't seem to make sense to have a launch duration of 2 or 3 weeks because this will not be possible to implement in practice. So we can create such an optimization problem. It doesn't need to be this way. It can be another ways. LAGO is flexible about that. But what I will discuss today is this particular optimal intervention package. In words, XOPT minimizes the cost of the intervention package while keeping the mean to be at least 0.8. In the presence of center effects, you could have an objective that the mean over the average over the centers of the mean given the intervention package is at least 0.8 while minimizing the cost. Or if you want to have different centers contribute different weights because, for example, they have different numbers of patients, you could make this a weighted average. Still, XOPT minimizes the cost of the intervention package while keeping the average mean to be at least 0.8. I will consider today Learn as you go or LAGO studies with 2 stages. This can be generalized, but that's not for today. We consider the situation where the stage 2 intervention package can be adapted or learned using the stage 1 outcomes. And the typical reason for a Learn as you go LAGO study is that if the study team decides that the intervention is broken, they want to fix it, and we should then allow them to do so. This leads, however, to a statistical complication, which should be the reason why statisticians didn't want to handle this type of data, because once you start adapting the intervention based on prior outcomes, the stage 1 and the stage 2 outcomes are no longer independent. And the practical statistical implication is that we cannot just condition on the observed interventions, because once we condition on the observed intervention, we implicitly also condition on a function of the prior outcomes. We then combine the stage 1 and the stage 2 outcomes and combine them together. We condition on all the interventions. We are conditioning on a function of the prior outcomes from stage 1, and that leads to all kinds of funkiness in the statistical derivations. Some notation. 2 stages, no early stopping. Each center is in 1 stage only. K stage can be 1 or 2. All these things here can actually be relaxed, but I don't do that today. And also this can be relaxed. The same number of patients in both stages makes it also a little easier. X1, the recommended multivariate intervention for stage 1, because LAGO is especially useful for multi-component interventions. X2N is the recommended multivariate intervention for stage 2. And we assume that is bounded. We have some say about that because we actually calculate this when this recommended intervention. I will say how. We are assuming that what centers do in practice, the actual intervention in a center J of stage K, that is a random variable that is not necessarily equal to the recommended intervention. And we see that in POLISA, in TaskPen, and in MapIt. The recommended intervention is there, but people actually do something else. So this is the actual intervention in center site J of stage K. And we assume that it depends on the recommended intervention, but not further on prior data. In other words, learning takes place through the recommended intervention, and that's all. It doesn't further predict the interventions that are taking place. YKN are the outcomes in stage K, 1 to N, the continuous outcomes in stage K, with YIKN the outcome of participant I in stage K. And from this notation, it follows that YI1N and YI2N denote outcomes of 2 different participants. This is an outcome of a participant in stage 1, and this is an outcome of a different participant in stage 2. We are currently trying to figure out about time-dependent outcomes, but that is not solved yet. The N superscript is needed in stages after stage 1 because the intervention in stage 2 depends on the outcomes in stage 1, and there are N of them. So we are not dealing with first figuring out where stage 1 goes? No, because stage 1 and stage 2 get large at the same rate. The main statistical complication with Learn As You Go LAGO, the recommended intervention is random and determined by some function of the prior data. We recommend that intervention. And it depends on what researchers decide upon looking at the stage 1 results. In terms of random variables, X2N is not independent of the stage 1 data, and that implies that also the outcome in the different stages are dependent. So we now assume that X2N depends deterministically on the stage 1 data. It's a pre-specified rule that we provide. For example, the decision rule could be based on means or percentages based on the entire stage 1 sample in a smooth, say differentiable way. Or the decision rule is based on an MLE calculated from the stage 1 data in a smooth, say differentiable way. And why is this? Well, if it depends smoothly on means or percentages of the entire sample and then goes to infinity, that this one will converge. And if this happens, because the MLE is usually consistent, we have to make sure that it is a setting where MLE is consistent, then this one will also converge in probability to a fixed design. This sets the stage for asymptotic inference. What happens as the total number of observations gets large, which seems reasonable in the Better Birth study and also in the current implementation trials. It's not unusual for these trials to have thousands of observations in each stage. And we will assume that the number of observations in both stages gets large at the same rate to obtain realistic approximation of what happens in finite samples. So here we are with the assumptions. The recommended stage 2 intervention is learned from stage 1. We provide that. The main assumption is that given that recommended intervention, the stage 2 data are independent of the stage 1 data. And so conditional on X2N, the stage 2 data are independent of the stage 1 data. And we are assuming that X2N converges in probability to a constant as the number of observations in stage 1 goes to infinity. In each stage K and center J, the actual intervention is a function of the recommended intervention, with this function being a deterministic, continuous, bounded, and center-specific intervention. We don't need to actually observe or know this function, but we do need to observe the actual interventions. This actually maybe sounds trivial, but in practice, this is not at all trivial. It turns out to be a huge effort on the study team to actually monitor what is actually being implemented. And we have found that one of the strengths of LAGO is that it forces people to figure out what actually happens in practice, which in and of itself turns out to be a useful exercise. We are assuming a generalized linear model and an outcome goal. In each stage K, the conditional mean of YIKN, the outcome in stage K of participant I, follows a generalized linear model with a certain error term. And in this presentation, I'm assuming that these are IID. I think if Professor Lucian has a question about it, I will have an answer to how I think it can be adapted, but that we haven't solved yet. So this is the known link function. So this is a generalized linear model, and simple link function would be the identity link. Then the mean would just be beta 0 plus beta 1 A1 plus beta P AP. And what we are going to assume is that the outcome in the end only depends on the implemented intervention and not on how that came about. So how it doesn't also additionally on the A's also depends on the recommended intervention. And a natural adaptation of the intervention is to estimate beta from the stage 1 data and obtain a beta 1 hat. That is a standard statistical problem because the first stage is not a LAGO stage. So this is a standard problem. We get a beta 1 hat and then we obtain the recommended intervention by solving this optimization problem, making sure that under the recommended intervention, the mean is at least 0.8 while minimizing the cost. There is one caveat. Sometimes this will not have a solution. If the study team has been too optimistic and within the bounds that are reasonable for X, there is no intervention that reaches a mean of 0.8, then we have been working on a shrinking method that shrinks the recommended stage 2 intervention towards the stage 1 recommended intervention in a smooth way, so that still we can show that the recommended intervention converges in probability. What is usually the hope of the study team is that somehow the intervention component composition X can be fixed so that the mean is at least 0.8. So the stage 1 has a recommended intervention X1, then we collect the outcomes. Then we calculate the recommended intervention based on the stage 1 data. We collect the outcomes. We are assuming that given the recommended intervention, the stage 1 and stage 2 data are independent and that the recommended intervention converges in probability. And then here are the generalized linear model GEE estimating equations. You see here the absence of the variance because we're assuming that we have IID errors. You would program this in R or in SAS by making sure that it is a normal distribution, like a dist equals normal. Then you get these estimating equations for the GEE. And so the LAGO design just estimates beta by solving these same estimating equations. In the case of binary outcomes, we've been able to show that those are also the MLE, even in a LAGO setting. In this case, it's a little bit more complicated because we are not making distributional assumptions on the errors and we do GEE. For consistency, we have shown that these estimating equations are unbiased. So we need to show that under beta star, the true beta, the expectation of the estimating equations is 0. So for this first part, that is standard because of a generalized linear models theory, because this is just fixed in advance. Here we can also just condition on the actual intervention in the particular center for each J. And then we get, this one gets out and we get this, and that is 0. However, this, so easy though it seems, it doesn't immediately get you consistency because again, we are not here dealing with IID terms. These terms and these terms are dependent through the recommended intervention. What we can still see is that there is no correlation between these two terms. And we can do that by conditioning on the later stage interventions. So we can deal with the two terms separately when we look at consistency. And then the next thing that you can see, this is standard because A1 is not learned. This we can also show that the variance of this goes to 0 as n goes to infinity. And so the variance of this goes to infinity, goes to 0. Oh, sorry. That is not yet that I need to be done. So the variance of each of these terms goes to 0. The expectation is 0. And then from Chebyshev inequality, we can show that this thing converges to 0 in probability. After that, it's almost standard to go next to the theory on empirical process theory that is presented in van der Vaart, and we get consistency. Asymptotic normality follows a different pattern. We do the usual Taylor expansion on the score or the score equations or the estimating equations around beta star, and this beta tilde is between beta hat and beta star, maybe different in each of the rows. And so as usual, we get this expression. This one is this numerator is kind of standard, is kind of easy because beta tilde converges to beta star. This numerator is not standard because you see here two terms, and these two terms are not independent. So what we did is we used an error replacement strategy. First, we replace this by the limiting intervention. So instead of aj prime 2n with a capital, we go to aj prime I without capital, with a 2 but no n, so it's limit. And we show that the difference between that and what we have here converges to 0 in probability. And after that, we look at this and we want to compare that with what would happen under the limiting intervention. So the limiting intervention is the AJ2 of the limiting recommended intervention. And because of the way we are assuming that the AJ2 depend on x2, and this converges also in probability. And then we construct outcomes under the limiting intervention. So we get the expected. Right. Somebody is not muted. Can they mute themselves? Thank you. So we have this expectation, but now the errors under the limiting intervention. And then we replace the errors because we're assuming that there are IID errors for now. We replace these errors here by the errors under the limiting intervention. And then from standard theory, we can get asymptotic normality, because once we have errors under the limiting intervention, and here also the limiting intervention, we are looking at a fixed design with the first stage, the non-LAGO stage that we already have, and the second stage, the limiting setup for the second stage. And that gets us asymptotic normality. That means we also have a test for no effect of the intervention package. If the null hypothesis is that beta 1 is beta 1 star, the Volt over likelihood ratio tests are asymptotically valid for any constant beta 1 star. An alternative test in a LAGO trial is just comparing means or comparing proportions. Because in a LAGO trial, we are assuming that how the learning takes place is through the recommended intervention only. And if the null hypothesis beta 1 star is equal to 0 is true, that means that however you learn doesn't affect the distribution of the stage 2 outcomes. So you can learn even from non-averages. You can learn however you want to. And still, under the null, that learning doesn't do a thing because whatever you implement leads to the same distribution of the outcomes. And that means a standard test for equal means in the control and the intervention arm is valid. If you have center effects, you need to take care of those. If you have other cluster effects, you need to take care of those. But that has got nothing to do with LAGO, because under the null, LAGO doesn't affect the distribution of your outcomes. That's only under the null, but the beauty of testing is only needs to be valid under the null. We have also looked at a power goal. To add a power goal, there are two options, and neither of them change the test at the end of the LAGO trial. We first estimate beta 1 hat from the stage 1 data, and then we can either look at the conditional power on the unconditional power and recommend an intervention that has sufficient conditional power or sufficient unconditional power. I will do the simulations really fast. I learned that at a conference. So what I'm just going to go like, look, 94, 95, 96, 94, 95, 96. Beautiful. Everybody happy? Questions? Thank you. And so we can also calculate confidence bands for the means based on asymptotic normality. And we can also create confidence regions for the optimal intervention package, because we can keep only package configurations for which the confidence interval for the mean outcome contains 80%. From a confidence band on, so we can also do confidence band for the mean, and based on those confidence bands, we can get the conservative confidence interval for the mean under the estimated optimal recommended optimal intervention by looking at the confidence interval in confidence band at that particular X. So here also, look, 95. What I want to do a little bit more is the Better Birth study, encourage the use of a checklist. That is a known working checklist, but not always implemented. People turn out to not even always wash their hands when they help someone giving birth. We looked at the proportion of checklist outcomes performed, outcome goal 0.8, and here we have the number of coaching visits and the number of launch duration days that we considered. This is called the LAGO table, also with a cost. And we have one center characteristic in the model. And you see here that we estimate the optimal intervention after stage 1. We estimate the optimal intervention after stages 1 and 2 combined. And the end, we use the data from all the stages to estimate the optimal intervention after the Better Birth study. And that is based on these estimated component interventions and doing this optimization problem with beta hat instead of beta. So this is a summary. It is the Learn as you go design to aim to find the optimal intervention composition and its effect. There is a dependence between stage 1 and stage 2 data. We used an error replacement argument and empirical process theory for the justification, a confidence set for the optimal intervention, a confidence band for the mean, and LAGO is currently used in several ongoing trials. We can have different number of patients per stage. We can have center-specific covariates. We can have as many stages as we like, pre-planned. We did also LAGO for binary outcomes and a power constraint. Currently, we are working on random center effects. This is joint work with Donna's postdoc Jingyu Cui, Donna Spiegelman, and Xin Zhou. We allow centers to contribute to more than one stage. Jingyu is working on that. Min Bui, my PhD student advisee, is also working on that in a different setting. Then there is look at study design, explicitly consider additional ways to choose the recommended intervention. We want at some point to be more flexible for binary outcomes. My former PhD student, Ante Bing, who graduated last year, or this year actually, this May, is working on that in collaboration with me and Donna Spiegelman to create an R package for the LAGO optimizations and for analyzing LAGO data after the trial is over. We are also working on confounding by indication. What if a center adopts the interventions in correlation with their potential outcomes? And we are allowing here for fixed center effects, and that turns out to deal with the confounding by indication if that is not time dependent. And that is joint work with Min Bui, a PhD candidate here at Boston University, Chris Longenecker, who is at the University of Washington in Seattle. We see him about every 2 weeks to make sure that we are keeping close to his extra CVD data application and things remain applicable, and also with Professor Donna Spiegelman at Yale. This is something that we just submitted. This is the one that I talked about today. This is the first LAGO paper, and thank you all for listening.
Thank you very much.
And I have 1 second left. I have 1 second left.
Excellent timing, and also very very clear and organized presentation. Thank you. So
Thanks.
next, let's have our discussant, Dr. Lu Tian, to present his thoughts on this work. So Lu, the floor is yours.
So let's see. I assume everyone can see the screen well.
Yes, thank you. Yep.
Yeah, firstly, I want to thank Jen Li for inviting me for this very exciting events, and also for Peter and Heidi for the organizing support. So I want to congratulate Professor Locke on her and her collaborators on this great work on LAGO. It is actually, I have to confess, the first time when I received after I received my assignment, I start to read LAGO, and it's a very exciting methodology. The concept of LAGO, learn as you go, is very appealing. It's a very special form of adaptive trial, but broader and more appealing than many other adaptivity we considered in designing and conducting a randomized clinical trial. It balances the cost and the effect, and also it's very flexible in adapting the intervention package. And the theoretical results are very interesting. The consistency and the normality was established elegantly despite of the non-IID nature of the data using the error replacement argument. I strongly encourage our audience to read the paper. The theoretical proof is very interesting, and the technique, I believe, can be used in many other similar settings for developing statistical methodology for adaptive trial. There are three key assumptions I just want to raise our attention. One is the LAGO method relies on a parametric model describing the relationship between the outcome and the components of the intervention. So in this regard, it's a little bit different from the conventional randomized clinical trial, where we oftentimes can estimate the average treatment effect almost non-parametrically. Here, we do need a parametric model. I think that is dictated by the nature of the LAGO I will discuss in the next slide. And also another assumption is the dependence across stage is the rule, the data-dependent treatment recommendation for the next stage based on the current data. So I think that is a very reasonable assumption. The last assumption is the optimal recommendation derived from the previous stage is unique in the limit. So in other words, if the sample size goes to infinity, there should be a optimal intervention strategy. I think this assumption is needed for establishing the theoretical results. So if I'm thinking about the conceptual origin of LAGO, I'm not saying this is the true origin of LAGO. Based on my humble understanding, the idea actually is very similar in spirit to some adaptive dose finding algorithm. I believe you also can find some other examples. But basically, in the dose finding study in phase 1 or phase 1/2 study, we sometimes collect the data and estimate the relationship between the dose and toxicity using Bayesian or frequentist model. And then we identify the maximum tolerant dose, like MTD level, according to the functional relationship you estimated. And then you are going to allocate the next batch of patients to the estimated MTD level. And then once the outcome becomes available, we are going to update the estimates of the functional relationship between dose and toxicity. And then we can repeat this process multiple times until we have enough confidence on the estimated MTD. So in the sense, it is similar to LAGO. We estimate some relationship and adjust and take action according to the according to our estimates. and then we iteratively optimizing our action and update our estimation. So of course, LAGO is operating in a much more complex setting. So I think it will be nice to summarize or to refresh ourselves about the basic objective of LAGO. At least to me, I think the most important objective of LAGO is to estimate the effects of intervention components on the outcome of interest. So there are other objectives for LAGO, for example, estimating the average treatment effect, as identify the optimal intervention package, and conduct conditional power calculations. They all can be achieved as a byproducts of the first objective, which is the estimation of the components effect. So because of that, I think that's why we need a parametric model to describe the relationship between the outcome and the intervention package components. So again, I just rewrite the model Professor Locke presented moments ago. So we need a model to describe the conditional expectation of y given x and z. And this model, I think, takes a parametric form. I'm wondering whether this model can be more non-parametric and more flexible. For example, can handle different type of outcomes, such as Cox regression for survival time, negative binomial regression for Const data, and maybe mixed effects model for longitudinal outcome. I think those extension, I think also Dr. Locke briefly mentioned in her presentation, will be very valuable for this method. Another issue I want to discuss is that if you think carefully, LAGO essentially, the methodology is essentially to establish estimating GE type estimating equation to estimate the mean model in the previous slide. So that means we need some variability in actual intervention received in stage 1 and 2 in order to estimate those model parameters. If everybody receives exactly the same intervention package, then we won't be able to estimate the effect of individual components of the package. So that means if every patient receives the same or very similar intervention, then, for example, according to the recommendation to the second stage, then there will be very little information for estimating the regression parameters we have interest. So I think the current LAGO trial, if I understand correctly, the variability of the actual intervention package received by the patients was induced by, for example, there are different centers. There is also center-specific effect. So I think in the worst case, if all centers are very similar or there's only very few centers, then probably we need to introduce some random perturbation about intervention recommended to different individuals in order to be able to estimate the components effect. Another issue I'm thinking is about identification of the optimal intervention package. It by itself is a very interesting mathematical problem. For example, under the assumed GE type or generalized linear effects linear model, it is almost like we want to minimize a cost function subjective to a set of linear constraints. Each of the components has a range, and also collectively a linear combination of those components has to be greater than a cutoff value, corresponding to the average response greater than 0.8, I think in Professor Locke's presentation. So this is essentially if the cost function is linear, this becomes a linear programming problem. If the cost function is quadratic, it becomes a quadratic programming problem. So, but in any events, it is essentially a map from beta to the components of the intervention package. it is should be a continuous map, but might not be differentiable, but it still allows to construct confidence band or confidence region for the identified optimal intervention package. But I think it's I think one complication I can think about is that if there is no or very little treatment effect, nearly under the null case, the optimal intervention strategy as defined might not exist. I think also Professor Locke briefly mentioned in her presentation. In that case, we might need to redefine the recommendation for the second stage by shrinking, for example, the new recommendation towards the initial intervention package. I think I'm curious about the impact on the inference by such adjustment. I'm wondering whether it is similar to the case that we are making inference for variance components in a random effects model, and the variance is very small or even equal to 0. In those cases, I think there's a phase transition for the inference procedure. Lastly, I want to emphasize a very interesting question after the LAGO is to estimate the treatment effect of the finally recommended intervention package. So after LAGO, imagine we know the relationship between the intervention components and the outcome, so we can design an optimal intervention package subject to the cost constraint. So we really want to know what would be the treatment effect of this intervention package, because this one is going to be used and recommended for the future patients in a larger population. So I think I suspect if we're only using the data to derive this optimal package, there could be potential bias, but I'm not 100% sure. Just like you use data to derive this optimal package, and then you plug in you plug in your estimate into the functional form to estimate the achieved treatment effect. I'm thinking the potential solution could be another stage or new study to estimate the average treatment effect non-parametrically, following the ITT principle, for example, allowing people have some adherence. And also maybe there are some analytical methods such as cross-validation for estimating this the treatment effect for this particular package. I think this is my discussion points. I really want to thank you all, and also I want to thank Locke again, and also Jen for the invitation. Thank you.
Thank you, Lou. Excellent points. So before we open the floor, just want to give Judith a chance to respond to some of the comments.
So thank you very much, Professor Lucian. You're spot on with your comments. Thank you so much. It's very interesting. So I took notes, so I will briefly address some of your comments. The dose finding, I think typically what people do is they find the dose first, and then, like you suggested in your last slide, do a separate study to estimate the effect. This is very similar to most multi-phase optimizations trials, where they have a separate phase where they tinker with the intervention and then test that last intervention in a separate trial that only analyzes the data from that last stage. So what LAGO does is we analyze all the data at the same time, but that doesn't prevent anybody to use LAGO just for the optimization phase of most, and then carry out a separate intervention study after. In terms of non-parametrics, I think that would be truly great. I hope that more people will start studying LAGO and try to tinker with the parametric assumptions. The type of outcome, I think I have a way to deal with that, to have it, I think the main complication is that the distribution of the errors is going to depend on the actual intervention implemented, especially when you talk about Poisson, for example. I think what can be done, but is this puts it back on my research agenda. This may be able to be done by using a QQ transform between the errors in the limiting intervention and the errors in the actual intervention. So instead of replacing the errors just by themselves, you could replace them by the QQ transformed errors of the limiting intervention. And then the difference between the actual limiting intervention and the QQ transformed intervention is going to be small. I have results on that from my work on structural nested models from causal inference, and I think it can be done, but I haven't done the math. But that puts it back on my research agenda. Thank you very much. Then in terms of variation, in the Better Birth study, we could only use 2 intervention components because there was a lot of multicollinearity. In other studies where LAGO is being applied is the Polisa Uganda, MEPIT Nigeria, and TASCPEN Zambia. There's a lot of variation in the inventor components. We look at monthly data and these models can be fit. So I think it depends a lot on the study and how strict people actually adhere to the intervention. But what we see is that people do so many different things that it led us to also work on LAGO with confounding by indication because we got scared. So that is that. And in terms of confidence interval for the optimal intervention, for the probability under the optimal intervention, what we've been so far doing is using the confidence bands. And the confidence bands should be good because that's what Chef Fay gives us. So we get confidence bands that are good based on the asymptotic normality result. And then if we look at the optimal intervention, that confidence interval, that confidence band at that optimal intervention should also be good. So I think it is conservative because we are basing it on the band, but I think it should be conservative. I think it can be made better by additional theory. So that puts that back on my to-do list. And yeah, the non-parametric, I really hope that people will pick that up. I am not sure that I will be the one doing that, but I think it will be totally interesting. I really hope that people will take that up. And who knows, I may become a non-parametric person and take it up myself.
Thank you.
Thank you so much.
Thank
These
you.
are excellent points.
Judith. Yeah, so we'll have the floor open for any more questions from the audience. So feel free to unmute and speak up if you have any burning questions. And also just want to mention that you also have the chance to email the speakers or the discussants for further discussion if you have more in-depth questions. And we'll also make the recordings available online. So if you have more questions after reviewing the recordings, also feel free to reach out to the speaker and the discussant directly.
Oh, and Judith, thank you for your contact information in the chat. That's very helpful. That's great. And I'm checking the chat to see if there are any questions. We do have a couple of minutes, so feel free. If you are on mute, you can type your question and we can take that. There's also an opportunity to submit your questions through the IBS community later on, but it's much easier now. So feel free.
I like answering questions, even if they're critical questions.
Absolutely. So let's see. I'm checking to see if anyone has their hand raised also. That's a feature we have today on this session. I'm not seeing anything at the moment. Jen, anything from your side?
I don't see anything yet.
Oh, I see a question just came in on the chat.
I can share my screen again because I have there on my, I think maybe the slides will be posted, no? So this is.
With your permission, we'd be happy to do that, yes.
I would be happy with that. So this is a second paper by Ante Bing. It's an archive preprint, so you can get it on archive. His first paper is in biometrics, so you know where to find it. It's also on archive. And then the other published paper on LAGO is here. So I think at the moment, everything that is ready for prime time is posted somewhere.
Okay.
The R package has not been posted yet because it's a work in progress. I predict that that will become available before end of year. And I would also not be surprised if this, where is it? Did I put that? So the work of my other PhD student advisee, Min Bui, so this work, I also predict that that will be posted on archive by the end of year, but not ready for prime time.
Okay, very
Yeah,
good. Thank you for
Thank
that
you.
question.
Yeah. Any other last minute questions? All right, since we are on the clock, I think that would conclude our session today. So thank you very much for the presentation and the discussion. That's excellent talks. And I definitely learned a lot about this LARGO study from your presentation. And also, I would like to thank everyone for joining us today. Now I'll hand it back over to Peter, the International Business Office.
Absolutely. Thank you so much, Judith, and thank you also for the discussion questions. Very helpful. As you all begin your weekend, I just wanted to send a general reminder regarding the opportunity to submit a contributed session abstract for the July 2026 International Biometric Conference, which will be held in Seoul, Republic of Korea, next July. So we will forward the link to the abstract submission site via the chat. And if you are a member of the IBS, you'll also see a message in the IBS members community shortly, if you haven't already. There are, of course, advantages to being a member of the IBS when you submit a session abstract or register. But please pass the information along regarding this submission period, because members and non-members are both eligible to submit. So we hope to see you in Seoul next July. We also hope to see you at the next Journal Club. We have, I believe, at least one more before the end of the year, if all works out, but we'll confer with the chair on that. But in the meantime, we hope you've enjoyed your Friday so far. And you may exit the session. This is the end of the session, and we will see you next time. Take care, everyone.
Thank you.
Thank you,
Thank you.
everyone.
Thank you.
Thank you.
Bye-bye.
Bye-bye.